Hello, everyone. I'm Director of Scientific Affairs at Altiview, and to start with, I would like to tell you a little bit about what Altiview does, and then we go into the core and the data that we can provide you. So what Altiview does is using ultrasensitive assays and AI-driven image analysis to provide deep insights to scientists that are trying to develop drugs for precision cancer therapies. How we do that and what is the main scientific community that we can help? It's like we are in the process of biomarker development. So we help the biomarker development during the development of drugs for cancer therapy. So there are a lot of publications that have shown that the use of biomarker during the development of a new therapy is critical, and they can increase the success or the approval of the drug. But they've also demonstrated that this can take much longer, and then it can really delay your trial. So this is where Altiview comes into play. So what we want to do is that we want to accelerate your research, but we also want to give better insights. And we can do that by using a technology, signal amplification, that allows to see more, but also faster, because the way we develop panels and deploy panels is a matter of weeks and not months like other technologies. And we can help you to see in situ, because we also have an image analysis pipeline that is AI-driven, that can really help you to understand this big amount of data that you generate. It's not just we provide very colorful images and then a pile of numbers, and then you need to make sense of it. So that's not at all what we want to do. We want to help you into the entire journey. So the way we do that, I told you, and I'll go very briefly about the technology. We use primary antibodies that are conjugated with DNA-barcodes. So this allows us to have all the antibodies applied to your tissue at once. So no cyclic staining, no stripping, no damaging to the tissue. So this allows us eventually to do also H&E, which is extremely important for pathologist annotation. So whenever you talk to a pathologist, they will always want to look at the H&E to annotate the tumor, to annotate areas that have to be excluded from the analysis, etc. So what is proprietary to Altiview is the signal amplification that allows you to increase the sensitivity of your assay. So you can detect also targets that are very lowly expressed in your tissue. We detect the signal by using fluorescent probes that are complementary to the DNA barcode that is attached to the primary antibody. And you see on this image only 4 antibodies, and this is done on purpose because this is not limiting us to a 4-plex, because we go up to 12-plex, and you'll see later. But we use 4 distinct fluorophores that do not overlap, so therefore we do not need to do any manipulation of the images at the end and do, for example, spectral imaging. Next, as I said, once we have developed the panel, we have applied the panel to your tissue, stained an image, then we can use our image analysis pipeline, which is deep learning based. Today, everyone says we have an AI-driven image analysis, right? So the difference is that we don't use AI just for tissue detection or for tissue segmentation or like compartmentalization, like to separate tumor versus stroma. But we use deep learning for each of the steps, including cell marker classification, which is extremely important. So imagine you have a tissue type that has a lot of, I call it background staining, but think of structures of the tissue that have autofluorescence. So like if you have collagen structures, for example, in FITSI you will see them. But then if you have an AI model that is not just looking at the intensity of your positive signal, so is not threshold-based, you will be able to identify those cells even in very tricky samples. And then what we do, as I said, we report insights to you. So we generate a report which is completely custom, and it really answers the scientific questions that you have asked at the beginning of the project. So Altiview can deliver these solutions in different fashions. So for image analysis, I mentioned already what we can do, and we provide this as a service today, but in a couple of months, this will be open also to any customer that is using our kits. In terms of the technology which we call in situplex, ISP, we have two ways of delivering it. So one is building kits that are transferred to the customer, to the end user, that can perform staining and imaging by themselves, and image analysis as well if they want, or we can provide our image analysis tools, or we can also perform staining and imaging ourselves. So we have a services lab. When we talk about kits that we can provide to customers, we have two different solutions. And this is applicable also to whatever is done in services. So what we call OmniView is a list of 24 biomarkers, and is an expanding list that can be combined in any fashion, up to an 8-plex so far, in a couple of months up to a 12-plex. So you can pick any of those targets, design your own panel depending on your hypothesis to be tested, and then we can put them together in a box that will be delivered to you, and you can apply the staining. This does not mean that we are limited to 24 targets. We have tested more than 300 targets, and any target that has an available antibody can be developed with this technology. And this is what comes into the category that we call UView. So you choose what you want to view, and any marker is on the table, basically. And we also don't only work with human, but we also work with mouse and NHP and other species. This is just an example of how you can build your panel. So we picked some of the combinations, and depending really on the target you choose, you can answer different questions, starting from a very simple one, like, is my tumor hot or cold, or to a more complex one, like, is my immune microenvironment reactive? Are there activated T cells? Is the tumor proliferating? Or are there TLSs, and what's the status of the TLSs? Or what type of macrophages are in my tumor? Are they polarized M1, M2, etc.? And all of that can be done very quickly. So again, you don't have to sacrifice time and make your development much longer, because everything can be delivered in 4 weeks, whenever you choose from those 25 targets. I also mentioned that we can work with any other target, and we do this by selecting first the antibody that is available, that usually we go with a clinically relevant clone, but if you have a clone, a proprietary clone, or if you have a clone to suggest, we can work with that one. We will start always with testing the clone in chromogenic assays, DAB, which is still the gold standard. And from there, we will develop a singleplex and then integrate it into your panel of choice in a multiplex panel. Then we can also provide some level of validation depending on your needs. The standard package we can provide is what we usually call a precision testing, which goes through reproducibility and repeatability testing. And then we can do staining and imaging, as I mentioned, and image analysis. Now, flexibility is something important, but we don't want to sacrifice quality when we want to have flexibility. So we developed a strong verification for the platform that we call OmniView. So we basically used the TMA that you see here on this picture that has 4 different tissue types. So you see tonsil, lymph node, colon, and melanoma. And you see that each of the tissues are in triplicates. Now, the triplicates are not just the same core, but are same tissue from different donors. So this accounts already for a little bit for the variability and the heterogeneity that you can find in tissues. Of course, tonsil is not the best heterogeneous, but this is very critical when you look at tumor types. So if you have 3 different melanomas or non-small cell lung cancer, they can be very different. So heterogeneity is also taken into account here. So we use this TMA, and what we did, this is an example of a panel that has been built for a specific customer question. So we took the TMA, we stained 5 serial sections, and then we correlated the intensity and density levels for each of the targets and also for different phenotypes in the same core in the different sections. So here you see an example of the comparison of a phenotype that is a triple expressor. So here we have CD3, CD4, and FOXP3. So usually when you look at correlations or you look at the coefficient of variability, you look at single target, because this is, of course, the more targets are on the same cell, the more the variability increases. So when you see values like 0.99 when we apply a Pearson or a Linz correlation, this is extremely high, especially because we are looking at a triple positive phenotype. Now, in this table on the right side, you see the values for each of the single targets, Pearson correlation, Linz correlation, and CVIS in percentages. And you see that when you look at CVIS, we don't reach 10% of variability. So that's very, very important. Second thing we did, we were wondering, like, what if we have markers that co-localize or are co-expressed in the same cell, and we combine them in the same panel, or we combine with markers that are on different cells? Like, is there any steric hindrance if we do this? So we built 4 different fourplexes that had common markers. Like, for example, panel A has CD8 in FITC, panel B has CD8 in FITC, panel D has CD8 in Cy5. And in the 3 different panels, CD8 is combined with different targets. So do we get the same number of CD8 positive cells if we have different combinations? And the answer is yes. So this is for cell densities. This is when we compare CD3 in the first graph, CD8, CD68, and cytokeratin. In different fourplexes combination, we also get very, very good correlations above 0.9. The next is intensity. So we have learned from experience and from talking to customers that intensity really matters, especially if you are quantifying functional markers. You want to know if it's lowly abundant, is it highly expressed? So intensity is really critical. And if your assay is not stable in terms of intensities, then whatever you're quantifying afterwards is not really reliable. So first of all, we have implemented a quantitative QC in our assay. So whenever you buy a kit, we will provide data on the intensity levels. And if you come back 6 months later and we need to generate a new batch of reagents, we are going to do again this type of analysis with intensities and compare to the previous one and make sure that the difference is within acceptable ranges so that you are confident to use it on different samples that belong to the same cohort. And also when we look at intensities, the correlations are above 0.9 for both Linz and Pearson. Next step is also, I told you we are not limited to 4-plex, and now I'm showing an 8-plex data. So what happens if I'm incubating my tissue with CD8 with other 3 targets or with other 4 targets or 8 targets or 12 targets? Am I gonna get the same type of data? And here is another example also, CD8 in panel in a 4-plex, in an 8-plex in round 1, and in an 8-plex in round 2. And again, the data are extremely comparable. So it doesn't really matter if you have your target detected in round 1 or if it's in a panel of 8 targets or even 12. So we are now launching a 12-plex panel. So we have done it for some time in our services lab, so we are confident that it's applicable in the field, but we are now releasing it as a kit and giving the opportunity also to customers to do it themselves. So this will allow more phenotypes analysis and a deeper understanding of the tumor microenvironment. And also for the 12-plex assay, we have generated a lot of internal data, more to come. I'm just showing one example of the reproducibility and repeatability. So again, we run 3 serial sections of tonsils that were interleaved in 3 different days. Here you see, again, intensities and densities, and you see how for each of the targets in a 12-plex, the data are very, very tight, so the variability is very, very low. And then if you look at the table with numbers, you can clearly see that for both intraday and interday, coefficient of variability are pretty low. The highest I can see is in intensity for FOXP3, which makes total sense because FOXP3 can have different level of variability, but CVs are very, very low. So just to summarize, we can work with you, understand what is your biological question specifically, and we go through a deep scientific consultation really to understand what is the objective of your study, and then we can build the right panel up to a 12-plex of the targets that are going to answer your question. Then we can provide staining and imaging, but even more importantly, image analysis. and give you the results in a report that already makes sense to you, is not just a lot of numbers. Thank you.